in the face of a worldwide crisis. We tried our best to unite the team. But we couldn't figure out how. As time passed and our attempts failed, we became desperate. There were only 8 months left to save the world. Time was running out. But finally, we re-entered the lab. And we prepared to tackle this project. Together.
Every year, over 80 tons of diclofenac, the most used painkiller in Germany, are consumed by humans. Concentrations of diclofenac in the wastewater can reach up to 1,500 nanograms per liter as a result of overuse, improper disposal, and unreacted excretion. Like other micropollutants, diclofenac can harm micro and macroorganisms living in water systems. A frightening example is that diclofenac excess in an ecosystem results in renal failure and mutations of the gills in the rainbow trout. Living in Germany, we recognize the dire concerns this heavily used drug brings to our local environments and realized with some research many wastewater treatment plants cannot adequately deactivate or separate these micropollutants before release. While diclofenac is a local problem to us, medical micropollution is not, and the gaps in smaller wastewater treatment facilities leave many areas of the world without sufficient resources to resolve these concerns on their own with current technology. We could no longer bear the thought of standing idle as this problem continues to plague the environment and its inhabitants, and used this motivation to guide our iGEM project. We hope to help combat harmful micropollutants from continuing to accumulate in water systems and create a cleaner future.
We wanted to come up with a viable way to combat the ongoing micropollutant problem in our water systems. It was important for us to find something that could be implemented not only in larger wastewater companies, but also by smaller facilities. Because current technologies like ozonation are really expensive, we needed a biotechnological solution.
Our project involves the transformation of the green algae Chlamydomonas reinhardti, enabling the chemical modification of diclofenac, resulting in its functional degradation. We will integrate genes for the enzyme lacase into the genome of our green algae, as well as perform a control by cloning the genes into the bacterium Escherichia coli. This will facilitate the production of these enzymes and secretion into medium, aka the wastewater, from wastewater treatment plants, where they can detoxify diclofenac. We split into 3 teams to best implement our experimental process. One team focused on producing proteins with our model organism called Chlammy team. One focused on doing the same with our control organism called E. coli team. And our assay team who performed the confirmation activity assays with both previous teams' produced lacases. The Chlammy team produced vectors with the genes for the lacases Barlac and Marlac via modular cloning. All level 2 multigene constructs included a spectinomycin resistance cassette for later selection. After transformation in Chlamydomonas reinhardti by electroporation, the algae were plated on TAP agar plates containing spectinomycin. Random colonies were picked and inoculated in fluid culture for screening. The cells were lysed and centrifuged to separate the debris from the supernatant containing the soluble lacases. SDS page and a subsequent Western blot were performed to find positive transformants. All lacase producing cultures were inoculated again in fluid culture and prepared for ABTS assay and HPLC assay.
The E. coli team purchased the vector, obtained through in vitro synthesis, from General Biosystems. First, 2 vectors, which include the gene for one of the lacases, Barlac or Marlac, a tag promoter, and an ampicillin resistance were transformed into E. coli. The transformed E. coli was grown on LB plates with ampicillin to select for the colonies that took up the vector. One colony was chosen and produced in fluid culture, with which the proteins were expressed. Production was induced with IPTG. After expression, proteins were purified with GST Cepheros columns. The concentrated protein was then ready to be tested with ABTS or HPLC. As a visible proof that the lacases were produced, SDS-Page and Western blot screening were performed. Because ABTS interacts with lacase in a similar way to the lacase-diclofenac interaction, a series of ABTS assays were performed to determine enzymatic activity. To begin, the optimal wavelength for ABTS, 420 nanometers, was confirmed via spectrometer. An assay was then performed to visualize the positive control lacase for Trametis versicolor at the two optimal pHs documented for Barlock, pH 4, and Marlock, pH 7, as well as its own optimal pH 5. In a 96-well plate, varying concentrations of the positive control lacase at different pH levels were added to 250 micromolar ABTS and run for 4 hours. Once produced, we ran the same assay with the manufactured Barlock and Marlock lacases, both intracellular and secreted, depending on what was produced, at their optimal pH and compared it against the positive control to confirm activity. HPLC was run first with diclofenac to verify standard retention time, then a positive control reaction with T versicolor, then our produced lacase to determine the product peak and verify oxidation of diclofenac by the lacase. To ensure efficient enzyme production, Chlamydomonas will be raised in a permanent culture in a bioreactor. To separate the green algae from the wastewater, we want to create a filter between the bioreactor and the wastewater basin that only allows the enzymes to pass through. The degradation products show no toxicity and therefore do not harm the environment. Safety is very important while working in the lab, especially when working with genetically modified organisms. We focused on working carefully with those organisms so that neither the team members nor uninvolved individuals inside or outside of the lab would have contact with them. As an additional safety measure, a strain of green algae that has no natural cell wall was selected and therefore has almost no chance to survive on its own in the environment. On the advice of Dr. Ehlers from the
Bundesamt für Verbraucherschutz und Lebensmittelsicherheit,
the Federal Office for Consumer Protection and Food Safety, we contacted the Landesbehörde, the regional state authority. In this digital conversation, we discussed the approval requirements and future implementation of a bioreactor with genetically modified organisms in a sewage treatment plant to better understand the steps required for making our project a reality.
Over the course of iGEM, we were able to produce both lacases, i.e. Marlock and Barlock, in the cytosol of the green algae Chlamydomonas reinhardti. Proof of the cytosolic expression of Barlock can be seen here, with the presence of a band at 70 kilodalton. Neither of the enzymes were expressed when fused to a N-terminal CCA secretion signal and a C-terminal HA RGS 8-his-tag or a SP20 HA RGS 8-his-tag. An increased copper concentration did not change these results. However, we hope that future teams can test more constructs, which we couldn't due to time constraints and COVID restrictions. Besides our main organism, Chlamydomonas reinhardti, we also wanted to produce lacase with the bacteria Escherichia coli. First, we transformed the plasmid PGEX6P1 with the gene of interest in E. coli BL21DE3 cells, and then we performed a growth test where we checked the viability of our manufactured cells and if they needed additional copper sulfate in their medium. On the gel, we noted the presence of our protein and saw no difference of growth between cells with and without copper sulfate. We started our protein production with E. coli BL21DE3 with optimal conditions determined by previous test expressions. After harvesting and lysing the cells, we purified our protein with affinity chromatography made possible by the GST tech. With a size of 89.3 kilodalton, we could find our protein on the SDS page at the level of a relative molecular mass of the marker between 75 and 100 kilodalton. When we did the last step of purification, we had our protein without GST tech. It was very visible on the gel with a size of 61.6 kilodalton and was detected on the membrane after doing a Western blot.
After both teams produced protein, they were tested in ABTS assays. Unfortunately, the lacase produced by Chlamydomonas did not show any activity. However, from E. coli BL21 demonstrated oxidation nearly as well as our positive control lacase from Trametes versicolor. The well A2, which contained the Barlac sample, very clearly reacted, and in the following normalized absorbance graph shows the positive control a similar enzymatic reaction rate to our produced enzyme, while the negative control shows no change. This data was reinforced by the HPLC performed later with a different replicate of the lacase produced by the same Barlac BL21 strain. Since diclofenac and other common micropollutants dissolved in the water do not change color as soon as they are oxidized by lacase, it is necessary to separate and determine the products by means of spectroscopic detection using the HPLC column. Diclofenac was used as a standard curve graph, and the average retention time for diclofenac was about 30 minutes. No obvious product peaks are visible, and while there was noise in the beginning of the graphs, it didn't change between the samples. A comparison was done at different time points over the course of an hour between our positive control and our produced Barlac, reacting with diclofenac. When oxidized with lacase, regardless of positive control or produced enzyme, the area underneath the diclofenac peaks appear to decrease at a linear rate until time point 10. At this time point, they become consistently low, suggesting complete oxidation and similar activity levels of both lacases, despite our very low concentrations in our produced Barlac. With such a low concentration to begin with, this sample reacting in such a clear way is suggestive that the produced lacase is not only effective, but possibly even more effective at oxidizing diclofenac than seen in the ABTS assay. Due to time constraints, contaminated samples and limited access to equipment, we were unable to perform replications to verify the results. However, the values are promising, reflective of what has been observed in literature and by our previous assays. So to review, the most promising construct was Barlac produced in E. coli BL21, according to a recorded ABTS activity and HPLC results. While we did try multiple strains as well as a different construct, Barlac, we recommend more replications with these to increase yield and confirm activity. It is also important to mention that the lacase produced by this construct is more stable in a more neutral pH than other available lacases, such as our positive control. While we recommend many replications be performed to confirm our preliminary findings and yield must be increased to make this option viable for real-world application, these results suggest an alternative option to expensive technologies that are not viable at smaller water treatment facilities at this time. But iGEM is so much more than just ordinary lab work. Unlike the traditional practicals undertaken by most students, we were challenged to completely self-organize a new, unprecedented project. We held many meetings in which we discussed the logistics of our experiments and were given constructive criticism and guidance by our advisors. And of course, lab materials do not grow on trees. To combat this, we initiated committees to organize ourselves, such as seeking out expert exchange for troubleshooting, posting regular updates over social media, or soliciting sponsorship from innovative companies to help fund our project. Through this, we obtained a lot of valuable contacts and gained more confidence by seeking advice from others. We operated like a small company, working together to create something greater than the sum of its parts. This was an incredibly valuable experience. We learned so much that we could never have learned through traditional studies. Moreover, the collaborative spirit of the competition could be felt in both the large and small interactions with other teams. Some were geared more towards team camaraderie and connection during the pandemic, such as the iJet video hosted by Team Aachen or the postcard collaboration with Düsseldorf. Some showed off to current works being done by teams in a more scientific light, such as the academic and Muggle journals organized by Team Maastricht. But all demonstrated how we could come together as members of the scientific community rather than letting competition keep us apart. We perpetuated this companionship by organizing many group meetings and information exchanges with our partners from Darmstadt and Stuttgart. They also had case-based projects focused on cleaning micropollutants, and we chose to create a page with useful advice for future teams. Through our discussions with other groups, we recognized that wastewater pollution is not just local to Germany. We organized international wastewater meetings with Finland, Czech, Switzerland, the US and other cities in Germany, where we discussed the different standards found within different countries and created a reference to aid our teams in future implementation efforts. With the understanding that wastewater pollution is a global concern, we knew how important it would be to keep the general public informed on the matter and to help better communicate how synthetic biology can help. Unfortunately, we weren't able to perform educational outreach in the more traditional ways this year due to safety restrictions, but we worked around them best we could with digital solutions through live streams, podcasts, surveys and Zoom discussions. Another key area of human practice is encouraging scientific literacy in children. We choose to do this by creating a workshop with a group of American students. We worked hard to make it as interactive as possible considering the distance and organized a local water treatment facility tour in addition to having students design a filtration experiment and report the observations. All teaching resources can be downloaded on our wiki for further educational purposes. Despite the encouraging preliminary results and all the hard steps we've taken this year to encourage change and foster understanding in our community, it is just the beginning of a long journey. Because of the circumstances, we had several new constructs we were unable to test, and many more we encourage other teams to try when access to the lab isn't so limited. After the meeting with the regional state authority, we finalized a testing scheme for a filtration system in our bioreactor and designed some pre-tests to verify our system would withstand both winter and summer conditions. These tests would be great next steps for future groups looking to carry our project ideas forward. We also toured the water treatment plant in Kaiserslautern, and during this we noted a gas supply already installed for the sand trap. This gas generator is directly beneath, so a supply to the bioreactor could be easily attached to make implementation much easier. Once perfected, the most viable and efficient algae constructs could be housed in this gas generator and start their career in saving the environment, one oxidation at a time.
For a cleaner future for you and me!